So I'm Chris Zahm. I'm a PhD postdoc working in the McNeil lab on cancer immunotherapies. And what we're trying to do is generate a better vaccine for the treatment of prostate cancer. So this is different than like a vaccine that you would get against hepatitis or a different virus, as in that those are preventative, and this is trying to treat a disease, not prevent one. So this will be for people who actually already have cancer, trying to cure it. Instead of using chemotherapies, we can use the person's own immune system to generate a response and fight off the tumor. And so what we're looking at doing is optimizing a vaccine that we've already had. We attempted to increase the affinity of the vaccine for the immune cells. This generates a greater immune response, so we have more active cells that should do a better job at fighting off the tumor. However, we found that that's not the case. These vaccines actually do worse at fighting off tumors in mice. This is because they express this molecule called PD-1. And that is a molecule that signals to your immune system that that's a self cell and to leave it alone. So it sort of prevents autoimmunity in the case of normal functioning immune system, but it prevents us from treating cancer as well. So the goal was to generate a model peptide that will give us activation of the immune system without inducing this PD-1 expression. And so we generated a panel of peptides again, looking at the affinity of those for the immune system, for the T cells. You can see here that we have a high affinity, medium affinity, and then two sort of lower affinity peptides. And when we stimulate those in vitro, so in a petri dish, what we see is that the high affinity peptide, the synthecal, generates PD-1 on the T cells, and that's sustained for as long as we can look at them in a dish. So it's sustained for 4 days out. Whereas these lower affinity peptides maybe don't generate quite as much PD-1 in the beginning, or they do, and then the PD-1 sort of falls off over time. So what we're thinking is that these T cells have been activated, they've responded to the antigen that we're showing them, they've expanded, and now they're not expressing PD-1. So these cells should go on to do a better job at fighting off tumors. They should produce a better anti-tumor response. Down here we're looking at T cell activation, so we can show that these cells are actually just as active in the beginning. So we get the initial response, the initial burst and amplification of the T cells. And then some other regulatory markers that do the same thing as PD-1. LAG3 and CTLA4 don't change, so that's not what's responsible for the effect that we're seeing with the anti-tumor response. And looking at PD-1 again on the active cells, so this is actually on the activated T cells, we see that PD-1 is increased in the high affinity peptide and not in the lower affinity peptide. So we think that actually, sort of counterintuitively, generating a peptide that has a higher affinity for the T cells is a bad thing when we're trying to treat tumors. So we have to find sort of the optimal peptide affinity that will give us activation without expression of PD-1. So that's where we're going.